I
did not record any of that.
Oh, all right. Well, want me to start again?
No, it's okay.
All right. Well, anyway, so this is the bee in the brain. All right. And this is not working. Okay, so the bee, the brain, and the biochemistry, right? So these are the three areas we're going to be touching on today. I'm going to start with the bee. I'm going to start with, then I'm going to go to the brain, and then we're going to talk about the biochemistry behind how they interact. And this is also part 3 in a series that I have done previously for the science department, and this is turning toxins into treatments, part 3. So now it's working, I think. So the honeybee, this guy got it. The honeybee, all right, so it's Apis mellifera, and its diet is pollen, nectar, and royal jelly for the future queens. They are naturalized, but they're not native. The honeybee, the Western honeybee, is actually not native to America. All right, it's been naturalized here, and it was introduced by the American settlers in the 1600s, and it's spread to all that bee has been spread to all the continents except for Antarctica for its economic benefits of pollination and honey production. All right, so within that bee, we have the worker bees, and the worker bees, they are the non-reproductive females, and the worker bees have specialized body type. All right, so for nectar collection, they have a straw-like proboscis, right? So that basically this long thing that they can take the nectar from the flowers. All right, they're a pollen collector, so for pollen collection, they have hind legs that have these pollen baskets that they can just take the pollen and kind of shove it into their legs, right? And then it also sticks to all their hairs, right? So that's one way that they can collect the pollen. The wax production is interesting. Can you decrease that a little bit? It's kind of in the way. Yeah, thank you. The wax on the underside of wax production. So they have wax glands on the underside of their abdomen. And so when the worker bees come back in, one of the things that happen is they'll eat nectar, they'll eat pollen, they'll get protein from that, and then they'll produce this wax, and that wax can be used to build honeycomb and stuff like that. And it's really interesting thing. I don't want to get into it too much here because it's not really the focus, but that's kind of a cool thing, right? Like they can produce it they've got these essentially cells that are producing this wax, and that wax can be used to create the honeycomb. All right. Thought that was interesting. All right. And then protection, they've got a barbed stinger with a poison sac that contains the bee venom. All right. Ooh. So bee venom. Again, now it's not working again. I don't know why.
It's just because it has to be on the, and if I touch something
okay.
else.
So then the bee venom, right? This is a mixture of bioactive compounds. All right. And melatonin is the pain-inducing part of one of the bioactive compounds. It causes blood vessels to swell and burst, and it's about 50% of the bee venom. We've got phospholipase 2A. This is the enzyme that leads to inflammation, and it destroys cell membranes. This also allows for the bee venom to get deeper into the tissues. All right. Also, the hyaluronidase is another one that allows the venom to spread. All right. The histamine, that's going to be normally what's produced in a normal allergic reaction to something, but it actually has histamine that it will give you, which is not good for those people that are allergic. All right, and that dilates the capillaries 12%. And then apamine is the neurotoxin that blocks potassium channels in the brain. And so guess what? That's what we're going to talk about. So apamine, This is a cyclic 18 amino acid peptide. All right, you'll see it's here. It's got a cysteine. I highlighted these two arginines within this 18 amino acid peptide. They are this blue one here, this R14 and R13. Those are to come into play later, so I'm just putting that. This is, so another part of this, these orange places here, these are disulfide bonds, I believe, or they could, I think they're disulfide bonds. And then this is an alpha helix. So if those of you who don't understand biochemistry type stuff, right? This here, this is an alpha helix. So, and then it's cyclic. So that's the whole thing is cyclic. And then you've got these amino acid groups. These are the side chains that come off of this amino acid backbone. All right. They become important in how they bind to certain things. All right. So it's the smallest, this apamine is the smallest known neurotoxin that's a polypeptide. All right. And it passes through the blood-brain barrier. So it very easily gets to its target, which is the brain. All right. And what it does is inhibit the small conductus calcium activated potassium channels. So SK channels. I'm just going to say SK channels from now on because, you know, right? That's a lot easier to say. All right. Everybody okay? Good. All right. So that's the main toxin we're talking about here. All right. Because that's the one that I did the research on and looked into. So apamine. Now we're going to talk about the brain a little bit. All right. So that was the bee venom. Now let's talk about the brain. We have explicit, so within the brain, we have memories, right? And how do we create those memories? Well, we have explicit memories, we have implicit memory, and we have working memory. So explicit memory are events that happen to you, right? If it's happened to you, it's episodic. If it's a general fact, it's semantic, right? Those two types of things. So, okay, it's a general fact, that's the semantics, right? Episodic means that happened to me, it was an episode, right? I can think about that in the future, right? Within the explicit memory, we have the hippocampus, the neocortex, and the amygdala are the three places within the brain. So you'll see over here, the hippocampus is very central. The amygdala is right in front of it. And the neocortex is the top portion of the brain here. All right, so those three areas are where these memories get encoded for. All right, we have initial storage of memories in the hippocampus. The neocortex stores long-term memories, and then the amygdala that adds emotional weight to that memory. Right, so those three things work together to have your explicit memory. Now, implicit memory, this is unconscious memory. This is motor memory. So when you go to ride a bike, right, this is where that's stored in the basal ganglia and the cerebellum. All right, and you'll see that problems within the basal ganglia can lead to Parkinson's can lead to problems where basal ganglia can lead to Parkinson's, right? Problems within the basal ganglia, right? So they've shown that. And then cerebellum, this is your more like your fine motor movements. So this would be, I would say, like basal ganglia is kind of like riding a bike. The bigger movements, the fine motor movements would be like playing an instrument. All right. So if you go to learn how to play an instrument, that would be coming from the cerebellum. All right. So you have your conscious, your unconscious. These are motor things, right? Cerebellum. Then your working memory. This is the shortest period of time. And your working memory actually comes from mostly just the prefrontal cortex, right? You have a left and right side. Your left side is the verbal and your right side is your spatial orientation. Right, this was the prefrontal cortex is essentially was the last portion of the brain to develop. All right, and that's kind of what makes us human. All right, and small amount of information for a short period of time. Right, this is what allows us to tend to get rid of the information we don't need. right? Like you have somebody talking in the background or whatever, and you can kind of just like buzz that out when you're not focused on it. That's the working memory. Okay? So we're going to talk about memories because we're going to talk about Alzheimer's. All right. And so what happens with Alzheimer's, it's the most common form of dementia. All right. Some of us probably know someone that has had dementia or had Alzheimer's. So we'll talk a little bit about that. It's beta amyloid plaques, right? These are peptides that build up. All right. And we'll talk about later how they build up and why they build up and what that leads to. But some of the early symptoms are forgetting recent events, right? I don't know, could be what you had for breakfast. I don't know. And disruption of attention. Those are very early symptoms of Alzheimer's. Okay? Very early signs. All right, we'll get into that a little bit more. All right, so I just wanted to find a couple of things here. First, synaptic plasticity. This is the control, this controls how well two neurons communicate. All right, and what happens here is we need this in learning and memory, right? Synaptic plasticity is key for that. And what we can do is essentially we can change synaptic strength, right? We do that naturally, right? Where we go back and forth from something being a strong connection to maybe a weaker connection, right? And we need that to learn stuff. Again, this is the idea of things fading into the background that you don't need, right? So that event that happened to you, maybe you look outside and someone's walking by, right? That doesn't really last, right? And so that's why we need kind of this long-term depression, or we need to modulate the synaptic strength. Okay? Now, long-term, so how do we do this? We have long-term attenuation, right? This enhances the synaptic strength. So you'll see if we have a potentinated cell, two things are happening. We're increasing the amount of molecules that are released in the synaptic cleft. And we can also do it by increasing the number of receptors on the cell that it's interacting with. All right. And we can have stronger connection. Right. But you don't want to have that strong connection all the time, because then that can lead to fatigue. Right. And that could lead to toxic environment within the brain. All right, because you have too many molecules that are there. All right, now, and you can also, so you have long-term attenuation, that's increasing that synaptic strength. You have long-term depression. This is reducing the synaptic strength, right? And it's essential for selective weakening, which you want to weaken some things and strengthen others. We need both for learning and memory. We've got to have both of those. So you got to have this back and forth between these two in order to have healthy memory and healthy learning ability. Okay? So it's almost like you got to go to sleep to be healthy, right? It's kind of one of the things that's there with that. All right? So that's plasticity. Now, so now I'm going to start talking a little bit about the channel that the aptamen binds to. So in the corner here, you'll see this SK. This is that SK channel, the small conducted calcium ion activated potassium channel, right? So what happens here is when it's activated, potassium can be released from the neuron. All right. And so we have 4 types of these channels. They're found in different parts of the brain. All right. They're activated when calcium binds to this molecule right here, which is called calmodulin. All right. And basically what happens is you have an excitatory event, right? You're telling your brain is telling your neuron to send a signal. All right. What happens is after that signal is sent, your brain, you have to shut down parts of that signal, right? As it moves down that neuron. You gotta shut down stuff. And so that's essentially kind of what happens here, right? You've got your calcium channel opens, right? Allows calcium in, sodium's being allowed in, right? And what happens is that depolarizes the membrane, right? So if you have a polarized membrane, that can mean I have more charge above and less charge below. There's poles there. That makes it polar. Good? All right. So there's a polarity, and what happens is calcium enters in. Calcium then binds to this SK channel and allows for potassium to leave to kind of reset it, right? So what happens is you end up getting, hold on one second. You end up getting an event. This is depolarization. This is resting here. We have resting here. We have depolarization. We have repolarization, and it goes back to resting. So send the event, whatever that event is, boom, depolarize. It's got to then repolarize to get back to where it is after that signal has been sent. Okay, I have another slide on this, but essentially that's what's happening here is we get repolarization, hyperpolarization, and we'll talk about that, but that's the responsibility of this channel. All right, and then they're highly expressed. Look at this. The second type is highly expressed in the hippocampus and the neocortex, right? And what were those responsible for? Anybody remember? They were responsible for, I need to go back. They were responsible for the explicit memories, right? So the neocortex, so we have a bunch of these channels in the place where we're getting the explicit memory. Good? All right. So they're highly expressed in those two areas, which is what we want. Well, that's what they do. All right. So this SK structure, it has 4 subunits that come together that make a tetramer, and they form a pore. So each tetramer has 6 transmembrane domains. 1, 2, 3, 4, 5, 6. All right, those are the membrane domains. There's 4 sets of these that wrap together, and these interact to create a pore. So this is where the calcium is going to go, right? Is right through this area between 5 and 6. There's a linker, right? It's called the pore forming loop. All right, between 5 and 6. And this is a selective filter for potassium. So what does that mean? It's selective for potassium. Well, actually, it removes water from the potassium to allow it to go through that. So the selective filtering, the filtering is that this pore removes water from the potassium, and that potassium can then go through, right? You'll see, it's hard to see here, but you got calcium binding to the chemodulin, and that's allowing for the potassium to go through and releasing that potassium, resetting and resetting. Good? All right. We'll come back to this slide in a little bit. Here's the hyperpolarization. So after hyperpolarization, we have the repolarization. After hyperpolarization is this refractory period down here. Okay, this is where it's repolarizing, and then boom, it comes down, it goes beyond, the threshold, which I've shown here on the board, goes beyond the threshold, and we have three types of after hyperpolarization. We have a fast type, which is 1 to 5 milliseconds. We have the medium type, which is 10 to 300 milliseconds. And then we have the slow type, which is 0.5 to several seconds, right? So what this does is it kind of slows down the ability for the next firing, right? So you want to have this refractory period so that you don't overstimulate. That's the idea, all right? You want to slow it down. So, and that's the neurotoxicity that I was talking about before, right? If it's overstimulated. And so the SK channels help mediate that. They help mediate the medium duration. So those SK channels are explicitly responsible for the medium one. So the 10 to 300 milliseconds. Okay. Now, with that said, so we have this channel. This channel is responsible for it. We have modulators that hit this channel and can do different things to it. We can increase that if we need to, right? So we have activators. We have things that will bind to this SK. Essentially what they do is they bind to that comodulin, and they make it more sensitive to calcium. So the calcium comes in, it opens up that channel faster and allows more potassium out. And you'll see here that what that does is reduces the cellular excitability, right? Increasing SK, which is this one down here. So you'll see that this hyperpolarization, this is longer, it's slower, right? So it's making it able to release more potassium and slow down whatever it's supposed to do, right? Whatever that is. Now, we've seen this, if we use these activators, it's called 1-EBIO, alcohol dependence, epilepsy, stroke have been used to treat this, right? To treat these, they have used these activators to treat this. I know, right? It's like, what in the world? Right. Good. All right. So we also have ones that inhibit it. You'll see Apamyn is here, right? And actually Prozac too. So we've got Alzheimer's, Parkinson's, depression. We have different types of inhibitors that will decrease that and make it more excitable, right? Make it quicker to excite again, right? And what that does is in a patient that has Alzheimer's and a patient that has Parkinson's, right? You want to be able to kind of bring back that ability to learn, right? If it ends up being too long, right? Like if we look here, if it's too long, right? Then that means you could get too weak. It could be too weak of a connection, and they might not be able to remember something because that connection's not there. So we're strengthening that connection by blocking the SK channel. Good? Everybody with me? All right. So how does it bind to the channel? Well, we've got, so the idea here is SK2 has never, how'd they figure it out? SK2 has really never been able to crystallize correctly, right, in itself. So when you go to find out, okay, so let me explain this a little bit. So when you go to find out the shape of this SK2, human SK2, you have to get it to crystallize and able to get an X-ray crystallography type of thing to it, right? You got to be able to see the shape. Well, SK2 has never been able to really be crystallize. So what they did is they took SK2 and SK4 and did a chimeric of them, and they were able to get that to crystallize. So they couldn't get SK2 to crystallize, so they created this other one that could crystallize. And then that's how to see where it bound, right? They knew it bound, right? They knew it worked on it because they could take apamine, put it into a mouse, and that was a model for Alzheimer's, and it would bring back some of the memory, bring back some of the not the memory, but they can bring back some of the learning from it, right? So they know it bound, but they just didn't know how it bound. So they created this chimera to figure out where it was binding. How is it blocking? Is it blocking directly, or is it blocking allosterically, which means away from the active site or away from the pore? Okay, so they created this chimera. It has properties of both. The properties were very similar to the wild type, right? You'll see here this voltage kind of tries to show that. We got wild type SK2, wild type SK4, and then the chimeras in green. And you'll see that they all have the same voltage profile there. So there's really not much difference between them except for minor things, but not where the apamine was binding. All right, so apamine binds to the 2 to 3 linker. Apamine binds SK2 and SK3 at the S3-4 linker. So this here is the linker between 3 and 4, those subunits that we talked about before. Here is the loop that creates the pore. All right, they also call it, it's the P loop, right? Here, and it interacts with this pore, right? Now, apamine binds to this linker, right? And you'll see that the places that it binds is that R13 and R14. Here and here, these arginines that I talked about. So what they also did within this paper is remove one of those R's and one of those arginines, and then it didn't bind. Right, so you need to have both of those R's there, and you also had to have this this is a phenylalanine, right? This F243 in order to bind, right? So it interacts, it binds. This is within the S3, S4 loop. It binds there, and then that blocks the release of the potassium through the pore. So it's binding here. That's shifting how these two things interact, how the P loop and this linker interact, and it blocks the pore. So it blocks the potassium from leaving. It's basically allosterically. So it's not binding directly in the pore, but it's binding outside of the pore and affecting the pore. Okay? All right. So how does this apamine help with the patients? Well, these alpha beta fibrils, or the plaques, the alpha beta plaques that are there in Alzheimer's disease, when that happens, you increase calcium levels. So if you increase calcium levels, you increase the activity, right? Which means I'm going to have more potassium leaving, which means more hyperpolarization, right? Which means less reactivity with the next cell and the next neuron, right? So too much calcium gets dumped in. Those plaques increase intercellular calcium. That increases the SK activity and then reduces the synaptic strength and can lead to memory deficits because you no longer have that strong connection, right? You have long-term depression, is what they call it, instead of the mixture of the long-term potention and the long-term depression mix. You get mostly long-term depression, which means it weakens that, and that's why they can't remember, or they struggle for words, or there's other things happening, right? Because these SK channels are a lot of the different places, but right, they can't remember events. But the ones that are really strong, those events, so a lot of times we see people that have dementia and Alzheimer's, and if you play them music back from when like they were in their prime, right? When they still had that, it'll trigger memories for them because those are things that have been potentiated for so long that it's still strong enough, right? But eventually that goes away with age, right? And that's the problem. So what we do with the Apamin is it binds to the SK channels, decreases that SK activity, right? Blocks the potassium from leaving, increasing the synaptic strength and reducing memory deficits caused by Alzheimer's disease. I think it's pretty freaking cool, right? I think it's awesome that bee venom, right? A part of bee venom can do that. Now, one of the issues with this is that Apamin right now is not what they call druggable, which means you can't turn it into a drug that you can take, right? You have to go get stung by a bee, right? Which is, who's going to want to do that, right? So what they have done, actually, if we go back to this, they have created, it's really cool. They're trying to create small molecule drugs that put this arginine at 13 and 14 in the correct spots. So they're using organic chemistry to build small molecules that could fit in that same spot that Apamin fits into. So I haven't looked too deeply into that yet, but I know that that's what they're doing. And eventually, hopefully, we'll get some therapeutic treatments for people that already have it, right? That can be taken. Good? I don't know. I think it's pretty cool stuff. Unless well, that's the thing, right? You're allergic, that's a bad thing. That's why they're creating these drugs also. And within some of these papers that I've read, they go through, this is how you synthesize it, right? They actually have put that out, like, okay, we made this and we tested, and some of them actually are starting to work, which is really, really cool stuff, right? And so that's how they are using, and this Apamin, they've known about Apamin since like 1985, I think. So they've known about this for a long time. It's just taking a while to get the technology to get there with it. All right. I probably went way too fast, but I can leave time for some questions, right?
Yeah, it just finally got settled.
I know. So, all right. Are there any questions? And yeah, go ahead.
The epilepsy part. So if a person wants to face an epilepsy, they can just go get stung by a bee.
No. So that would be an activator. So epilepsy some of the drugs that they're using right now for epilepsy are there's a lot of side effects, and so they don't like to put people on them. But this would be the opposite of a bee sting, right? The bee stings are the ones that are doing they're decreasing purple. How this is, the blue is the normal, purple is decreasing SK activity, right? The red one is increasing that. So think about it, right? Epilepsy it's almost like, what do I want to say? It's almost like it gets caught in a loop, right? Like the neuron gets caught in a loop, right? So what happens is this will allow the calcium to bind stronger and open this up more. So this one EBO-IO is one of the drugs that they're using to treat epilepsy. So this has already made it to a point where this is druggable, right? It gets inside the membrane and intercellular, so it's inside the membrane. It gets there, it modulates it, it binds to that calmodulin. Let me see if I can find it. So it binds here and then allows this to be more sensitive. And if this is more sensitive, it opens this up faster, so less calcium is needed to open up the potassium, so it releases more potassium, so it drives it deeper and will essentially shut off that loop, right? That loop of the neuron not stopping. Good?
Yeah.
All right. Hopefully I answered that correctly. Or
There's a
to your,
question here from the audience.
okay.
Would you happen to know, or is the relationship being studied of Alzheimer's and depression as comorbidities? I would assume that one would exacerbate the other.
Yes, I would agree with that. I know there probably is some comorbidity there, and that's also because, again, you have, if I go forward here, right? Yeah, I'm not sure how they relate, but I'm willing to bet that there are some other SKs. This is mostly SK2 that I'm talking about. So there could be other SKs in there, or these potassium channels that are in different parts of the brain that would then, yes, also be, so I agree, yes, 100%. I don't know of if anybody is using bee venom to treat that, but I mean, the Prozac still hits the same inhibitor. So I don't know if anybody's using Prozac to treat Alzheimer's. I don't know if that's been done or not. And again, I don't know if it's the same, and if it's the same style of inhibitor. I mean, the same style of SK, right? The same type. So it could be that it's a different SK somewhere else. It might not be SK2, it might be SK3 or 1 or 4, right? So hopefully that answers your question. I know I had a couple more questions inside here, in the room, so, okay.
I know you might maybe describing, like, surveying anecdotal stories, or maybe even
Can the question be repeated for the interpreter, please?
Oh, yes, so I'm sorry, it wasn't, oh, go ahead.
do I just need to speak louder?
Well, the
Yes.
speaker-
Yes, please speak louder.
Okay, sorry. So I don't know if you have the answer to the question, but I was wondering if any of your papers talked about whether, like, anecdotally or performed, like, the experiment, because I don't know how viable it is, of like what concentration of bee venom had been utilized either in animals or maybe even anecdotally, people who pursued like homeopathy, or like homesteading solutions, because I know bee venom is used for people who suffer from chronic pain. So like fibromyalgia, plaque psoriasis, or even arthritis. They'll use it localized because the histamine, it just kind of like trips your immune system into helping lessen the pain of the nerves that are firing and stuff like that. But I'm wondering, like, to detect Apmin, even being able to do this, was there like, you know, did they have to take a poor mouse and like inject it with a crap ton of bee venom, or like what are the avenues to understanding the Apmin can help with these specific illnesses?
I believe the Apmin was, I think it was something in the nanomolar to picomolar range. So it was really small amount. So for these studies, they didn't need a whole lot to study it. And I think, and remember, it's a small amount within, it's only like what, 3%, I think. Yeah, 3% of the overall bee venom is the Apmin. So again, for these studies, they were really, I saw papers that were like in the nano to picomolar range. So it wasn't very much. But I don't know, like
spread in the body enough, even though it's such a small. Is it just a very potent compound even though it's so small in the body. It's like the equivalent of like one bee sting's quantity.
Oh, I
would
don't,
have that 3% in it.
well, yeah, that's gonna have 3% of apamine, but I don't know. I didn't get into the quantities like per bee sting or anything like that, so I'm not sure. Right, did they ever, well, I'm sure they did. I just don't know how much for per bee sting or anything like that. Question.
I know some people with Parkinson's and depression. How would they get access to something like this? Because, and they're allergic to bees.
Right,
So that's,
so
is
that's,
it possible?
I think right now. There are some strong candidates. I think one was like, I think it's UCLI or UC, I'll write it down. This is a whole class of inhibitors. So, it's UCL 1684 is the class of inhibitors. That's being very specific, but that's the class of inhibitors that they're designing the drugs right now that they're this class. So I guess it's I don't know if class 1684, I'm not sure what I don't know anything about that. I just know that that's the class of drugs. So, and I'm not sure how many have gotten to that point where they've been able to get it away from the rest of the bee venom, right? And just make these small molecules. That's the idea, is making these small molecules now that they have an idea of how it works and how it binds. Now, and this paper came out 2025. So this paper,
Oh, wow.
yeah, just came out. And so people are now starting to look into this class of drugs. So, yeah, that's all I can say, because I'm not sure. A lot of that would be if a company, the problem is if a drug company is on this path, that's all gonna be kind of probably classified right now. If anybody's searching for that, that's the issue.
That sucks.
I know, but it shouldn't be, right? I believe in open source, just about everything, and I think, but again, I know there's people working on it, right? And I'm sure you can also, if you want to read the papers, you can go in the references, just, I'm sure Dr. Cardona can get you the PowerPoint and then you can look through those. I think the reference I'm talking about, is, I believe it's this one here, Samantha Castle. It's this one, right? And I think it's that one. And I think that's the major one that I was looking into. And small molecules, see, that's the small molecules that they're creating. So that's the paper that you can look into if you want more of that. Good? Good. You're welcome. Any questions online from the people who are zooming in? Nope.
Not seeing anything, but I did accidentally send the wrong survey, so I just submitted the correct one online. If you don't mind using that one, if you already submitted the previous one, that one's from last semester, the one that you looked at. The people here, you have the correct, talking about people online, have the link from last semester. So if you submitted the survey, please click on the updated link. And if you have any questions, let me know. You can raise your hand or submit a question on the chat.
Okay, I know we have one more question inside the room.
So you said that they're building the apamine and they have to move the, what's it called,
Arginine?
Yeah, they have to move it to their place. So my question is, obviously this is like a biological molecule in the bees. Even louder? Okay, I already go being so loud. Okay. Is it like a very tedious process to make this polypeptide specifically? Or like, is that what's slowing them down? And why is it different from other peptides that are easily replicated in lab? Like, why is it so hard to move that arginine?
Well,
Like, would you think maybe in like how many decades would it take us to make this a really speedy process?
no, I mean, the process is getting faster and faster, and it's a high throughput. It's essentially a high throughput to find out these molecules. You can use, there's a lot of techniques in how to do high throughput screenings. So they will go through and they will make this and see if it'll bind mathematically, if it'll bind correctly. They think it will, and then they'll screen so it'll go from a whole bunch of these down to a few, and then you screen those to see if it has an effect. And some of them have already, but now it's just, all right, getting them up to the level of being able to be druggable, right? So to being able to create a drug and be able to put it into form to where you could take it, digest it, or take a shot, right? Have a shot of it, right? So sometimes what they're doing is I've saw some of the papers that they were isolating the apamid and then just injecting that directly. So that might be able to get it away from the idea of having it as an allergenic, right? To making it, I guess you would say, hypoallergenic. But yeah, I mean, I don't know how long it would take to do that, but I mean, they're currently creating them, so I don't know, 3-5 years maybe. I don't know, right? That's one of those things where, of course, it's going to be marketable, right? You're going to there's enough people in the population to make it marketable, to make it profitable for the drug companies. So that's another thing you have to think about. Do enough people have the disease? I know it's a weird thing to think about, but do enough people have the disease to be able to make it marketable? I know it sucks, but that's part of it. Do we make this drug? Do we not make it? Can we make it cheaper and get it to more people? Is it stable? Right? That's the other thing. Is it shelf stable, or does it need to be stored in a refrigerator? Right? Can you take it to a third world country and have it survive long enough to get the population, right? Those are the things that need to be thought about when you're creating these treatments, right? So, I mean, you can go talk about Sabin and Salk and vaccine development and all that type of stuff too. But yeah, so I don't know how long, but they're working on it, which is the cool thing. I think it's hopeful. All right. Good. Answer your question? All right. Very good. All right. I don't think I have anything else.
No, nothing else here.
Nothing else there? All right. Thank you, guys. Hopefully you learned something. I don't know. I think bees I think they do have a secret life, right? This is a secret life.
Well, thank you for the presentation, Professor Norris.
You're
And next week on Wednesday at 3 o'clock in this room / on Zoom, we'll have a story about the truth about bees and what bees actually need saving. Since, as we learned today, the honeybees are not native and they don't, in fact, need saving. So we have an entomologist coming in to tell us about which bees do need saving.
Very cool.
And so thank you for everyone that's here and online. If you haven't done so already, there's a QR code on the desk here and a link on the final link that I submitted onto the chat is the correct one for this event. Thank you so much, and let us know if you need anything, if you want the presentation or the recording of this event, just email me at ccardona, which is CCARDONA @ccbcmd.edu. Thank you.
All right. Thank you.